Hello, my name is Riley Rafferty, and today is April 16th, 2026. In this presentation, I'm going to summarize Giorgio Bernardi's 2019 article titled The Genomic Code, Persuasive Encoding of Chromatin Structures, and a Solution to the Non-Coding DNA Mystery. The non-coding DNA mystery refers to the question of why so much of our DNA does not code for proteins. For many years, scientists thought that only genes that coded for proteins were important. The rest of the genome, sometimes called junk DNA, didn't really have a function. However, over time, researchers began to realize that this DNA must have some sort of function, because it makes up majority of the genome and is conserved across species. Bernardi's review proposes that this non-coding DNA actually plays a critical role in what he calls the genomic code. This is different than the traditional genetic code that determines how DNA sequences are translated into proteins. Instead, the genomic code refers to how DNA sequences influence the structure and organization of chromatin, the complex DNA and proteins that form chromosomes. One of the key concepts that Bernardi discusses is the concept of isochores. Isochores are large regions of DNA that have relatively uniform base composition, particularly in terms of the GC complex, which is a portion of guanine and cytosine bases. Bernardi shows that these isochores are not randomly distributed. Instead, they correlate with specific chromatin structures, gene density, and levels of gene expression. For example, GC-rich isochores tend to be associated with open chromatin, which is more accessible for transcription, and therefore more active in gene expression. In contrast, AT-rich isochores are more linked to compact chromatin, which is less active. This suggests that the sequence of DNA determines how the genome is physically organized within the nucleus. Bernardi also provides evidence that these patterns are evolutionarily conserved, which means they have been maintained over very long periods of time. This supports the idea that the genomic code has an important biological function rather than it being random. Based on this evidence, Bernardi concludes that the non-coding DNA is not junk, but instead plays a fundamental role in shaping chromatin and regulating gene activity. The genome contains multiple layers of information, one that codes for proteins, and another that influences how DNA is packaged and used within the cell. In my opinion, this article significantly advances our idea of the non-coding DNA mystery. It provides a compelling explanation for why so much of the genome does not code for proteins, showing that these regions are used for structural and regulatory functions in the cell. I think the idea of the genomic code is specifically important because it shifts how we think about DNA, not just as a sequence of instructions for building proteins, as a system that also controls the organization of genetic material. Overall, Bernardi's works helps bridge the gap between DNA sequence and chromatin structure, offers a more complete picture of how the genome functions. It highlights that non-coding DNA is essential for organizing the genome and regulating gene expression, which is crucial for understanding evolution, development, and disease.